Q&A session at the end. And with that, it is my absolute pleasure to introduce our speaker today. Dr. Serena Hung is the Chief Medical Officer at a therapeutics company. She has experience working in drug development for ataxia. Prior to that, Serena was a practicing neurologist who saw ataxia patients. The ataxia community's resilience inspired her to join the National Ataxia Foundation's board of directors, where she currently serves. And Serena wants to engage the community in advancing drug development for ataxia. And with that, I'll turn over the floor to Dr. Hung.
All right, thank you, Celeste, and thank you everybody for joining. I just wanted to, thank you for, taking an interest in clinical trials and learning about it, because, I think, there are more and more clinical trials in the ataxia space. So, it's actually, very important that, our community stay informed. So with that, I just want to start by introducing myself. Let me advance my slide, which I am unable to. Okay, here we go. So as Celeste mentioned, I am a board-certified neurologist trained in movement disorders. And in my training, I have seen ataxia patients. And so that was actually the beginning of how I got interested in ataxia. So I'm currently based in the United States, in the Boston area. So I, share the misery of the cold. I was in clinical practice and was a site investigator in clinical trials at an academic medical center until 2011. So at that time, I was seeing patients, and some of my patients had ataxia, and I was doing clinical trials, mostly in Parkinson's disease at that time. And since 2011, I've been doing drug development. And my responsibility as a drug developer is designing and conducting clinical trials, as well as strategic planning. What that means is that I help the companies that I work for, decide, what to develop drugs in, and what are the risks, and how do I think about, advancing drug development within the company. So also, as full disclosure, I own stocks in a number of pharma and biotech companies. So today's objective, I would like to provide a high-level overview of measures to minimize risks to study participants in clinical trials. And this actually encompasses a lot of different areas. So, if I'm moving too fast, if things are not clear, please go ahead and put it in the Q&A, and we can address that at the end. So I want to actually start out by pointing something out that, on the FDA website, this is actually what they say that they're supposed to be doing. So I'm not going to read the whole thing, but essentially what they are outlining is that any drug company seeking to sell a drug in the United States have to test it. And then the company has to send the agency the evidence from these tests to make sure that the drug is safe and effective for what it's intended to be used for. And then within the FDA, there is a team of experts, basically physicians, statisticians, chemists, and a whole lot of different disciplines. They will review the company's data and propose the labeling. And this review is supposed to be independent and unbiased. And at the end, they need to establish that a drug's health benefits outweigh its known risks, and that if they are able to prove that, then the drug is approved for sale. So front and center, they point out that, with any drug, there are some risks. And so what they intend to do is to make sure that there's more benefits compared to the risks. So I think this is actually something important to keep in mind, that with drugs, we can't have a complete safe ride in a way. So this is a little plug for the PrepRARE page on the NAF website. if you want to learn more about drug development, I would highly recommend looking into this page, From Research to Reality, the 5 Steps in Drug Development. And that page actually go a lot more into, what it entails over the course of, the life of a drug, basically. So using this diagram, one thing that I actually wanted to point out is that, as you can see, in all phases of drug development, with all of the clinical trials, safety is front and center. Also, especially in the earlier phase trials, safety is the primary purpose. So you can see the phase 1, safety actually is the only thing that we're supposed to be looking at. And then as we actually move along in the development of this drug, then you start actually thinking about, maybe efficacy. One of the key things to understand is that in the beginning, when we're looking at a trial with small number of participants, it is actually very difficult to determine the benefits. And so the early phase trials are designed to be mainly safety studies. So I'll start out, from the beginning, before a drug even enter into human clinical trial. And we call those preclinical studies. And what that means is basically they're laboratory studies. And we use animals. And so these animals are given the potential drug for a long enough period of time to understand the potential safety signals. So what I mean by long enough time is that there is actually regulation that if you want to, in the beginning, if you want to dose a patient or a participant 14 days, you would have to have dosed animals for 14 days. You can't skimp on that period of time because you want to actually understand with that duration of dosing what could potentially happen. So if you want to dose somebody for a month, you need to have enough, dosing, for example, in a rat for a month before you can even talk about designing a trial for a 1-month duration. I also want to talk about what do we mean by safety signals? So safety signals could be abnormal physical examination findings, and we do physical exams on animals. You can actually look at abnormal x-rays, or labs. And, these signals basically tell us that the potential drug may be doing something undesirable. It could be actually quite minor, but you want to know, especially at this stage with the animals, what we want to understand is the range of safety signals to watch out for. Also, what happens in animal studies is that we actually dose animals starting from a lower dose, and then we actually ramp up the doses in stages. And what happens is when any drug, if you get to a high enough dose, you're supposed to see some safety signals. Because that's actually an indication that the drug is actually active biologically. And so what we do is we actually look at the dose that are safe in animals, and then we sort of dial it back and to a dose where it doesn't do that. But we actually already established the outer limits. And then after we actually establish this dose range that are safe in these animals, then there are actually formula to calculate what could potential human doses be. Now, also in these animals, because we actually take samples from the animals, including after they're sacrificed, it is actually very helpful to understand the mechanism of particular safety signals. Why is it happening? What, along the pathway of the mechanism is it happening? And that way you can understand, is there anything that you can actually monitor for even earlier before these safety signals happen, for example? Or is there anything that you can do to help prevent it from happening? And with all of this information, it will help inform human study design. And also another thing that I'm going to go into later on is that there is something called the Safety Monitoring Committee. And it consists of people. So, knowing what actually happened in the animals would inform what kind of people do you want on the safety monitoring committee. So for example, if I see, liver enzymes getting high, so I may include a liver expert in the safety monitoring committee and so forth. So next, after we did the preclinical studies, I want to talk about what does this kit take to start human clinical trials. So first of all, we have to identify the most appropriate study design and write a study protocol. A protocol basically is the study plan. Everything that happens within the study has to be contained in the study protocol. And that's the document that we always go back to if in doubt. And some of the key things that are included in the study protocol would include, what is the study population? Who are the people that are supposed to be included in this study? What study procedures? So, what do we do? Do we do an X-ray? Do we do an MRI? How often do we give drug? And how long do they stay, within the clinic and so forth? But also importantly, what are the safety monitoring procedures? For example, what are the signs that we look out for? What are those specific blood tests we need to be doing? And so forth. And we write it all down in clear detail so that, sites can actually look at it. Everyone can look at this document and actually understand what they're supposed to be doing. So next, we have to actually draft the informed consent form. So informed consent is, basically a process by which the study participants will get told what are some of these potential risks. And so this informed consent form is supposed to be written in language that is easily understood. You don't need a medical degree to understand. And if you do, that is a poorly written informed consent form. And actually, there are specific guidelines as to how we should be writing the informed consent form. There are actually external parties that are going to be reviewing it to make sure that it is actually clear, that it contains information that needs to be in there, and that it's written in a way that it's easily understood, so that when people sign up, they know what they're signing up for. So the next part to think about, starting a clinical study is we want to identify study sites. And why is it important? It's because clinical studies are actually pretty complicated, and it requires expertise to carry out the study. So we want to identify study sites that have the experience, have the expertise, have the personnel, and the bandwidth to conduct the study so that things don't get overlooked and that people are taken care of. And we then compile all the data available in what we call a filing package. And basically, all the animal studies that we have done, from the earliest of studies showing that it worked in some animal models to the toxicology studies. And also, for drugs that actually have been used for other things before, but now you're repurposing for a different population, you have to include any data from any prior studies in human. And then you also need to include manufacturing information. So this is something that, as a drug development personnel, not really deeply involved in manufacturing, sometimes I forget, but it's actually, we're talking about a pill, we're talking about a solution that we're putting into people. So it's got to be safe. And there's actually very specific standards that are required that a company has to fulfill in order to actually put this drug into people. And you have to demonstrate to regulatory agencies that you do fulfill all of these requirements. Then we actually file for approval to the FDA with all of this information. Now, in my talk, I actually use the FDA a lot just because I'm US based and a little biased. I work, with the FDA quite a bit, but all the other regulatory agencies do the same thing. They look at essentially the same packet of information, and they have to make their own determinations. And then also simultaneously, we have to submit this documents to what we call an ethics committee or institutional review board. So it's IRB EC or IRB. And I have a slide that actually will go into, what these things are and how they function. But the ethics committees and institutional review boards usually do a review simultaneously with regulatory agencies like the FDA. And in order for a trial to start, you have to get both the regulatory approval as well as an ethics committee approval. So I want to actually bring it back a little bit to talk about what the FDA is supposed to do. And actually, like I said, the other regulatory agencies also do the same thing. They evaluate new drugs before they're sold. So, this is actually what we mean by, giving them all the data that, all from the different clinical trials that are done. And they have to have all of the data so that they can understand, whether the drug is, doing what it's supposed to be doing. And then also the other aspect of what they do is that they have to provide doctors and patients the information that they need to use the medicines wisely. And this is actually straight from the FDA.gov website. And how they communicate this information is through the labeling process. And the drug label is basically a compilation of all the information about this drug so that doctors and patients can understand, what was shown before and what are the risks. And the FDA actually has very strict procedures to basically say what can go into the label and what cannot, and what should and should not. So they look at the benefits and risks prior to approving new drugs. And that's actually probably pretty understandable, but also after the drugs have been on the market, they continue to look at, the benefit and risk. And primarily at that point is looking at the risks over longer term dosing. So as promised, we're going to talk about the IRB, Institutional Review Board, or Ethics Committee, EC. So the purpose of this group of people is to assure, both in advance and also during the clinical trials, that appropriate steps are taken to protect the rights and welfare of humans participating as subjects in the research. And they use a group process. They basically review, the research protocol and all of the related information. So for example, I talked about the informed consent documents. They want to make sure that it's written in an appropriate language. And also the investigator brochure, which is basically a handbook for the site investigator to look at if they actually wanted to look up information. And that's actually written in language that is more medically oriented. And so the IRB actually has to look at all of that and make sure that things are okay. They also look at the research protocols and they actually provide feedback on study procedures. So if, a certain risk is identified, they would actually look and make sure that in the protocol there is appropriate procedures to monitor for it, for example, and the monitoring is appropriate and that, people are not too burdened. So that's actually is another thing that they do to protect the welfare of humans, of participants. So actually one thing that is unique about IRB or EC is that it has membership from very diverse disciplines. So, it's not just scientists. You get MDs, PhDs, that's very typical, but you also get, other healthcare provider, nurses, nurse practitioners. But outside of the medical profession, you also get people like clergies, you get ethicists, you get lawyers, and sometimes you get community members. So basically representative from the community to sit in this committee so that they can actually provide different perspectives. Also, another thing that is important to know is that IRBs are not government agencies. So that's different from, the regulatory agencies that are government agencies. So the next thing that I want to talk about is, how do we actually safeguard participant welfare within the study design? So one thing that actually is very important to any pharmaceutical physician, as well as actually any doctor in the clinic, is that a good clinical study needs to have a very clearly identified purpose. What questions need to be answered with this study? Because if you design a study with not a whole lot of purpose, you're basically just exposing people to risk without actually understanding further what the drug does. Or if it actually gives you murky answers and you can't make decisions out of it, that would not be a good clinical study. So the study procedures are designed to answer these key scientific questions, as well as monitor for safety. So we only include procedures that help answer either the scientific questions or safety questions. We are supposed to be very judicious about what we include, because anything that we include in the study takes up people's time, takes up effort, and sometimes it exposes people to potentially more risk. So we actually have to be very careful about what procedures we put into the study design and, in turn, the study protocol. Now, the safety monitoring will incorporate information obtained from, preclinical studies, as well as, information on medicines in the same related class. What I meant by that is that sometimes, there might be a drug that actually already is farther along in, let's say phase 3 clinical trial, or maybe even approved. And so there was actually existing information on that drug. And now a new drug in the same class, but a better molecule, potentially better molecule comes along. We actually have to think about what was seen before within the same class, because some of the same issues may actually be seen in this new, formulation of this new version of this medicine. So we actually have to incorporate all of that and then design what safety monitoring procedure needs to be in there. So some of the pretty basic safety monitoring that you would see very frequently would include, doing vital signs, for example, temperature and pulse, weight, that sort of thing. You might do basic labs, so blood work, such as blood count, chemistry, kidney function. I didn't put in there, but, urinalysis sometimes. And you want to do electrocardiogram to look at, the heart rhythm and so forth. So these are actually very, common things that are included in clinical trials, and that those procedures are no different than what you would normally get at a doctor's office during a clinic visit. So one thing actually I wanted to point out is that in clinical trials, we actually have something called stopping criteria. And, the reason for having stopping criteria in clinical trials is such that, there are actually bars that, you can't cross, lines that you can't cross. So you have to pre-specify the stopping criteria in the study protocol. And how you determine what the stopping criteria is based on prior knowledge about the particular compound, as well as the known class effects. And so you sort of draw a line in the sand, and if this line is met, then the study is required to halt for investigation as soon as possible. And once actually these stopping criteria is met, you have to notify by rule, you have to notify, a group of people, and I'll talk about who these people are. Some of them we already talked about, for example, the IRB. You have to actually immediately tell them that, yeah, we have to stop the study because of, stopping criteria XYZ is met. And you look at, you also have to notify other people, and I'll go into, who these other people are. And DSMB is Data Safety Monitoring Board, which is actually a group of independent experts that actually look at the available data. And they're not, they actually operate pretty independently of the sponsor, i.e. the drug company. And they actually have a lot of power because if they think that, the if there's enough safety concern, they can actually recommend to stop the study altogether. So they are pretty powerful, and I'll go into how they function. Now, like I said, once you actually meet the stopping criteria, then an investigation would happen. And the action to be taken actually would vary depending on what they find. So, I'll give you an example. For example, if something happened and, but it turns out, it actually happened in a participant that was receiving a placebo drug, or placebo, sorry. So then cannot be attributed to the drug. And so, if that was the case, then the study could continue. But depending on, what it is, how mitigable means how fixable it is, um, the trial can be stopped permanently, or the study may resume, but, you have to make changes to the protocol, maybe more safety measures, maybe at a lower dose. And then if it's unrelated, the study may resume. So one of the other things that I want to talk about is how safety monitoring changes over the phase of development. So here, I think just, as cartoons, you see that, in the early phases, there are fewer people involved. But as you actually move along in the clinical development process, once you get to phase 3, it is not unusual that, hundreds of people are involved in a phase 3 trial. So what happens over the course of the development of a drug, there's more and more information gathered because there's more and more exposure in human, both in the number of people exposed and also the duration, of the drug exposure. So with that, you actually know more, and you can actually adjust what you do in terms of safety monitoring. So what typically happens is that in the early phase, in phase 1, some phase 2s, these safety monitoring procedure, it typically is more intense and very detailed because at that point, you don't know as much. So you want to err on the side of caution. So then we might draw blood a little bit more because we didn't know if something's going to happen, when it would happen, but we don't want to miss it. So you typically find that in the early phase studies, there's actually more intense procedures done in safety monitoring. Now, with the later phase studies, it really depends on what we see in the phase 1 and phase 2, for example. So if actually the phase 1 and phase 2 are done and there's not a whole lot of very concerning safety signals, then we may actually be able to lessen some of the safety monitoring intensity. Maybe instead of drawing blood every week, we may be able to draw it every month, or from every month going to every 3 months. But there are times when actually, if you actually find something new in your phase 1 and phase 2, and you want to make sure that it's thoroughly understood, then you may actually continue these intense safety monitoring in your phase 3. So it really depends on what you see over the course of the studies of these drugs over time. Now, I put in post-marketing here because it's actually something that we don't talk about as much. And within the ataxia community, we don't have necessarily a whole lot of drugs that, are approved that requires a post-marketing study. And so phase 4 studies are studies that are done after a drug get approved. And usually they are done, number 1, to understand longer term safety and efficacy. Because in a post-marketing setting, usually you actually take a lot more people. The inclusion criteria is not as strict, so you can involve a whole lot more people. But when you involve more people, some of the rarer things might come out. So in certain drugs, actually, the regulatory agencies would require that a post-marketing study be done. And even if a phase 4 study doesn't happen, I just want to reassure you that, we actually still, as drug companies, we still actually have to report every year to the FDA. What have we seen? What have we heard through our safety internal company safety system? We actually have to report all of that stuff into regulatory agencies, and every year they would actually say, okay, you can continue selling this drug, or no, we need to actually, understand more, or you need to stop. Like, they can actually do things even after approval. So I think that's actually, at least to me, that's something that is comforting, because you know that someone's always watching. So what happens during a clinical trial to minimize the risk to participants? And there's actually quite a bit happening during the clinical trial. So I talked about the informed consent. So before a participant signed the informed consent to participate, the investigator at the site has to review potential risk with potential participants, and they have to go into detail. And so for those of you who have been involved in clinical trials, sometimes, the informed consent is quite long, but the investigator is supposed to go through that in detail and answer any question that anybody has, both the participants and a lot of times their care partners, family members, and so forth. Everyone is to understand, on these risks, and that they're comfortable with it. After signing the informed consent, then the investigator has to carefully review the inclusion-exclusion criteria. And that is to make sure that the participant is suitable to receive the study drug. So, for example, a lot of times in the exclusion criteria, there are things that, stipulate that, if you have, active liver disease of such severity, you shouldn't be actually in this study. So those are the guidelines that basically tell the investigator, you want to select the right participants, and you also don't want to expose people to risks that we're not as sure about. And then also the investigator has to do a physical examination. The lab test, imaging tests have to be done prior to randomization and administration or taking of the study drug. So if someone actually, get a blood test and it shows that their kidney function is not normal, there's actually different degrees of not normal, but if it's severe enough, then the participant, or the person who actually signed the consent form, will not be able to participate. So at each visit, the investigator or the site coordinator will review any adverse events or serious adverse events with participants and a lot of times the care partner. And you have to do, all the physical examination, the lab tests that are specified by the protocol. Now, if there is actually an adverse event or a serious adverse event, there might be additional tests that are required to actually understand what is happening and also monitor people until the symptoms resolved. So, if there are such tests that are required, then we will have to do that during these visits. And actually, in between visits, the participants should contact the site staff in case there is some potential adverse events. Sometimes what I do when I was a site investigator is I tell people to err on the side of caution. If something is weird and you just don't know what's happening, I tell people to just call, because, sometimes it's kind of hard to figure out. And it's always good to let the site staff know and understand whether further workup is needed. So the site staff usually keeps pretty close tabs, or are in close contacts with participants, for that reason, just in case something happens. So if adverse events or serious adverse events happen, there are actually certain things that have to, that will be triggered, certain events that will be triggered. So, for example, the FDA and other regulatory agencies have specific requirements regarding timing of reporting of the SAEs, because, you want to know as soon as possible so that action can be taken. But even if it's, regular AEs, we have to actually report them regularly. You have to notify IRBs. You have to notify other investigators. And then site investigator has to follow the participant until the resolution of these adverse events. And depending on the nature and the frequency of these occurrences, sometimes the trial may have to be halted, the protocol has to be changed, or the trial permanently stopped. So these are some of the people that provide oversight during a clinical trial for safety purposes. And I'm going to go a little quicker because I'm looking at my watch. Okay, so the first line is always a site investigator and coordinator because they're always in direct contact with the participants and care partners, so they know. And they are, they can actually get the information direct from participants. And then medical monitors, or what we call sponsor medical directors, which are basically pharmaceutical physicians like me, we would review participant data regularly. But I look at it in a blinded fashion, so I don't know who's getting placebo, who's getting active drug. And I have to actually look at it very regularly across all clinical trial sites so that I can detect trends. And I also would communicate with site staff as needed. So for example, if I'm spotting something and I'm wondering if certain things have been done to, work it up, for example, then I would actually communicate with the site staff. Now, this data safety monitoring board, they will regularly review the data, but they look at it in an unblinded data so that they actually know who's getting placebo, who's getting active drug, so that they can actually have a true view of what are the potential benefits and what are the risks that we know thus far. And we talked about the IRB. So we have to actually notify the IRB in a timely manner. And I talked about the FDA and other regulatory agencies, based on the reporting, and they can also take action to demand changes to the trial. So a little bit more about the Data Safety Monitoring Board, and they're a committee of experts, and they're responsible for reviewing clinical trial data on an ongoing basis to ensure the safety of study subjects and also, more importantly, the validity, integrity of the data. Remember when I first said, a clinical trial has to answer questions. If the data are not valid or what we consider good, sometimes it raises questions as to whether, the question has been answered. So the Data Safety Monitoring Board actually has the responsibility of looking at that. But more importantly, they actually look at the safety data periodically, usually like maybe every quarter or something, depending on the phase of the study. And they actually look at it and determine whether it's too much risk. They may actually raise questions and ask the drug company to do things to further evaluate these, potential risks. They have full access to unblinded data. So that's actually something very important because that way, they actually can get a true view. They are independent experts. They don't have any vested interest in this particular treatment. And if they start having any potential conflict of interest, they will be kicked off of the Data Safety Monitoring Board. Usually they would actually tell the drug companies, say, I can't serve in this capacity anymore, but we do periodic checks to make sure that they are still actually independent, truly. And they actually they're pretty powerful. They can actually recommend the company to, continue the trial. They can tell them to modify it, or they can actually ask the company to stop a trial. So they're actually pretty powerful. So this is actually a little peripheral, but the Scientific Advisory Committee or Steering Committee, or probably terms that you've heard, but this is actually a group that is separate from the Data Safety Monitoring Committee. And they provide oversight to the clinical trials over the whole trial. They don't get to look at unblinded data. They basically want to understand and supervise the progress of the trial. And they actually also look at information from other sources, and they communicate the progress of the trial to relevant parties, and also advise on publicity and presentation or publication of all aspects of the trial. And typically, there's a independent chair. And one thing I want to point out is that, especially in rare diseases, representation by a member of the patient community is increasingly common to provide oversight to the trial. So there are a couple of things that I wanted to talk about, and in a way, I don't know where to put it, but I think they're important. So, observational study. So, usually an observational study is a study where participants are followed for a long period of time. They do get, assessments, both physical examination, neural exam, blood drawn, sometimes CSF to, understand biomarkers and so forth. But there's no drug involved. So even though there's no drug treatment given, observational studies have some unique considerations, and they may actually have risks. So things to think about: confidentiality. Just by the virtue of participating in an observational study means something, right? So if I'm looking for patients with a certain disease, and if it leaks, that's not good. There's disclosure risk, but one of the things that actually is, I want to highlight is genetic testing, because we want to actually understand for genetic illnesses what happens even before people start getting symptoms. So genetic testing is necessary so that you can understand whether the person has or does or does not have the gene required for the disease. And a lot of times, especially in the pre-symptomatic stage, this information is very sensitive, and not only to the outside world, but even within a family, right? So there's actually these considerations of, do you tell, who can you tell? Do the participant even want to know, for example? So those are all of these considerations that are very unique. And one thing for observational study is that even though there's no drug given, it still requires IRB or ethics committee approval. So they actually will still be looking at the merit of the trial, and also particularly look at, are there procedures in place to safeguard the the bullet points, the issues that arise from the bullet points above, and that participants are sufficiently protected. Now, if an observational study is a multi-center trial, they may have an external monitoring body, but that's actually is variable. And for external monitoring body, the composition also varies. It's not as uniform as within a typical clinical trial that involves a drug. So another thing, I think this is my last slide. Want to touch on data privacy and security. Something that, in our day-to-day life, we kind of think about it, but maybe not too seriously. But when you're thinking about a clinical trial, there's actually a lot of potentially sensitive information obtained in clinical trials. We also understand that, in order to advance science, we want to share data, especially with other researchers, to advance the field. And a lot of clinical trial participants actually express the desire that, they want their data to be used in a meaningful way to advance drug development in the field to help future generations. But we need to think about the balance of data sharing versus privacy of the individual. So I put the GDPR here. It's just to remind me to talk about, there's this law, basically, General Data Protection Regulation in the EU. And that has actually changed quite a bit of how things are done whenever we involve EU participants in clinical trials. There's no equivalent per se in the United States, but within individual states, there are rules and regulations that, we have to comply with. And the intent is that, we follow these guidelines so that if we share data, we're sharing it within predefined parameters that are acceptable to the regulatory agencies. So the things that we do to minimize risks, in that regard, is that, number one, we actually have to include in the informed consent what are the potential data sharing strategies. So, for example, a lot of times in clinical trials, we may actually ask people that, whether we can actually share the data, with larger databases. And if, this is the intent, we actually have to put it into the informed consent to say, please give us permission to do that. And these are things that IRB and ethics committees, they look at this very carefully to make sure that things are pointed out and that they are highlighted so that it's not hidden somewhere. So other things that are also done or keep in mind is that personally identifiable information are never incorporated into the database. So what that means is that, at the site investigator know who the participant is, and they would, they would know, the birth date, zip code, where they live, and a lot of information. But there is actually limitation as to what actually gets put into the database. And there are actually a lot of things that don't get put into the database, or they're blinded, so that I, as a pharmaceutical physician, I can't tell. Even if I look at the database, look at the individual lines, I can't tell who that person is. And that's, by design. Also, importantly, I think we do that, with our daily lives as well. But this is actually sort of like on steroids. The data systems and the databases are required to have measures to ensure confidentiality and security. So, something that you all know, like everything has to be password protected. Everyone has their own password, so that if something goes wrong, you can actually trace, where did it fall apart, for example. So those are some of the, and there are actually rules and regulations surrounding that as well. And we actually have to demonstrate that our data systems that we use comply with the rules and regulations. So there's actually quite a bit of checks and balances that go into these this design of these systems and the implementation. So in conclusion, I just want to, highlight that drug development does carry risks. And ultimately, drug approval require the benefits of the treatment outweigh the risks. And clinical trials are an opportunity to understand the benefits and the risk of a particular treatment. And in order to minimize risk, careful study of the drug starts with animals. And then during the course of clinical trials, there are multiple layers of safeguard in place to minimize the risk to participants. So any questions, please put them in the Q&A, and I look forward to hearing your questions.
Thank you so much, Dr. Hung, for such a fantastic overview. I feel like the curtain's been pulled away and got a peek behind how things work in the clinical trials that make the drugs that keep people healthy and safe. As a reminder for attendees, if you do have any questions for our speaker, you could drop them in the Q&A window at the bottom of your screen. You can click it to access that. While we're waiting for some questions to come in, I have a few of my own to start. On your slides, you mentioned adverse effects, but also serious adverse effects or events. Could you clarify the difference between those two things?
Yeah, so usually, so basically we have to document any little thing that happens in a clinical trial if it's out of the ordinary. And for the things that are just a little bit abnormal, we consider them adverse effects, adverse events. Now, the serious adverse events actually have a pretty, they have definitions. So I'll give you some example, not to be morbid, but death is one. Hospitalization is one, and anything that is life-threatening, but the person didn't die, but that's also an SAE. And then there are actually criteria. It's more better defined in the cancer world of, for example, your lab test has to be 5 times the normal limits, for example, in order to be considered serious. Whereas if it's 3 times abnormal, then it's still just an AE. So there is guidelines, but there are also some gestalt.
That makes a lot of sense.
Mhm.
And as a follow-up to that, if someone is in a current clinical trial, they've had their informed consent, they're an ongoing participant, but they now, perhaps after listening to this talk, have some questions about the safety procedures involved in the study they're in, who should they direct those questions to? Is there a specific person involved in a trial
Mhm.
who is responsible for answering those kinds of things?
Yep. So usually, just speaking from practical experience, the clinical trial coordinator is probably the best person to start. So these clinical coordinators, trial coordinators are usually, very experienced in doing clinical trials. They can have a healthcare background themselves. A lot of them are like, for example, nurses. And so, and also you can get them on the phone much easier. And you call them, tell them what's happening, and or you tell them the question, and then some of the questions they may be able to answer themselves. And for the ones that require medical expertise, for example, then the coordinator will then talk to the site investigator and get back to the participant usually. And also actually, if it's just a question, that's easy, but we always tell people if it actually something that looks serious, and go to the emergency room. When you're in a clinical trial, you can also go to the emergency room. So yeah, we just want to make sure that you know things don't get out of hand, that people are safe, that people
Mhm.
err on the side of caution. So long answer to start with the coordinator.
That makes a lot of sense. And thank you for that tip about going to the ER. I know sometimes things, it's like in the moment, it can be very difficult to make a decision. What's the right thing to do here? So that's very helpful to know. So
Mhm.
we have another question. You mentioned a little bit of this in your presentation already, but what does safety have to do with a drug's approval? You mentioned early on how like a regulatory agency needs to weigh the risks and benefits. Can you speak to that a bit more?
Yeah, so this is actually one of the things that I think um, we sometimes actually as physicians we forget too, is that you know depending on what the benefit is of a drug, people are willing to tolerate different levels of risks. So I'll give you an example. There could be a drug that is super effective, work really well, but it causes an itch. That's considered a risk, right? And so then the question is it an intolerable itch, or is it an itch that, goes away after a week? And once you get through that, you can actually continue. So that's why actually from a risk standpoint, it actually encompasses a lot. Now, one thing that we want to make sure, when we're developing drugs and when the FDA looks at the risk versus benefit adjudication, we want to make sure that the risks are not unjustified or intolerable. And there's actually a lot of discussion, especially among patient communities, about you know, what are some acceptable risks, knowing that with every drug, you're going to have some risks. I mean, it could be simple as, I have some nausea. It's a risk. And it might be tolerable to some people, it might not be tolerable to others. And so, who actually gets to make that determination? is actually a very interesting um, question. But one thing that is actually, I see the regulatory agencies doing more and more is they actually ask patient communities, like if it's something that is fatal, maybe you're willing to take a little bit more risks. And so ask the patient community, what is it that what risk are you willing to tolerate in order to get that potential benefit? So that's actually there's some judgment call in terms of this risk versus benefit, but ultimately, it is a balance. Um, you know,
So it must be a lot to juggle. And I know these are ongoing conversations. So thank you for
yeah.
summarizing that for us. We have an attendee who's asking, a lot of clinical trials seem to be happening in the United States.
Mhm.
Uh, Speaking generally, of course, do studies reach out to international participants? Do they tend to recruit locally? Are international folks even eligible to participate in clinical trials based in the US?
Yeah. So it really depends on, um, the face of the study and also the strategy. So usually when it's early phase, you're dosing very few participants. Sometimes it's actually in healthy participants. So you only do it in a single site and you want to actually make it as simple as possible logistically. So that's one thing. But once we actually get to a multi-center, then the question becomes, um, are there enough participants in other countries, outside of the US? I mean, the reason why a lot of trials are done in the US is because there are a lot of people in the US. So, a lot of times, just because of the sheer size of the population, and you might start in the US, but it really depends. So, for example, for genetically or genetic diseases, you go where there are patients because when there's a founder effect, there's actually certain areas of the world that they just don't have any people. So those are some of the considerations that we would make to decide where we would be opening clinical trial sites. And there are times when the US doesn't get to participate because they just don't have either the infrastructure or the population to participate in a trial. So it really depends. Now, if you're thinking about, the question is about crossing borders to participate, generally that's not advisable because number one, and I will tell you from a company standpoint why it's very difficult. Number one, there's regulations. So, actually some countries actually don't want you to do that. And then practically, there's also the insurance issue. I'm like just keeping it very real because usually in the insurance clauses, it will actually, be specific to a certain country. So if you come from a different country, insurance doesn't cover you and doesn't cover the site. So that actually is something that, logistically make it very challenging. Now, there are times when, especially if it's a rare disease, it may be able to actually apply for special provisions for people to travel, to another country, but that actually requires, special procedures to do that.
Of course. And we are at time, but just one final question. We have an attendee who, after hearing this talk, wants to learn more about current clinical trials. As a professional in this field, are there any resources or websites that you would recommend for someone who wants to learn more about ongoing clinical trials for ataxia?
Um, I think the NAF actually has a list of trials that are currently recruiting um, uh, participants, and they actually also have a little blurb about what the trial is about, what is the study drug, and what are they looking for. Like, for example, is it a phase 1 trial? Is it a phase 3 trial? And Stephanie put in the chat,
Yep.
the website. But that's actually probably is the easiest. I mean, you can also go to places like clinicaltrials.gov, but that is, I hear complaints that it's actually a pretty hard to navigate
It could be a bit tricky.
website. Yeah. So, if you have time, you want to play around, That is also another source of information.
We try to collate as much as we can, but sometimes it
Yeah.
takes a little bit of time to update our end of the site. So if there's a trial you find that we don't have listed, please let us know. But with that, if everyone could join me at your individual Zoom screen, thanking Dr. Hung sincerely for a fantastic overview of safety in clinical trials. We really appreciate your sharing your time with us. And thanks to everyone here for joining us for this past hour to learn a little bit more about what goes on behind the scenes to keep us safe as we advance ataxia research. I hope you found something useful from this session, and I wish you a wonderful rest of the day, wherever you are in the world. So good morning, good afternoon, good evening. I'll see you at the next PrepRARE session.
Thank you. I hope it was useful.
Yeah, take care, folks.
All right.